Thanks, Stephanie, and thanks for inviting me here. It's been a long day to get here, I have to say. Hopefully I'll try not to fall asleep, and hopefully you won't either. But you're more than welcome to, really. And I'll try not to keep you away from the important things which I hear clinking around outside. So I guess about 5 years ago, I really got interested in the whole pharmaceutical sciences business. And I have to say I was very struck. I do different things in different time periods, but I was very struck when I started doing this, is that it's important, as we were discussing with the students a little while ago, to ask the big questions. And here's a big question in my mind, what really happens when I take a drug? And what I concluded when I started looking into this for the first time is that we don't necessarily have a particularly good idea. The whole notion of Ehrlich's magic bullet of one drug, one target to treat one condition has served us to a point, but it's not necessarily what goes on in the majority of cases. And I'll try and convince you of that as I go along. So that's really the question we're going to address. But before I do that, I was asked to sort of meld in some other things that we do around professional development and the like. And so one of the things that we discussed already a little with the group was the idea of asking these big questions, which I already mentioned. And in the spirit of that, there's Hamming, and many of you know the Hamming distance, but he actually posed a lot of really interesting, I think, ways to do the best science. And one of them was to ask the big questions. And so, we may not actually be answering them, but we feel, at least in our lab, that we're trying to address the big questions. And we actually wrote 10 rules that adapted that, the spirit of Hamming. So I'm not going to talk about most of these, but we already had a little discussion about, one big question is how, I believe that the way we disseminate science is going to change dramatically in the next few years. And how that takes place and how we can improve the comprehension of science is something I'm very interested in. In fact, I probably spend as much time doing that now as any of these other things. And then other questions, so the one we're going to address is this big question here. But it turns out that to answer that question, there are other big questions that actually feed into it. Obviously, the question we're asking, we're particularly interested in neglected diseases because they're neglected primarily because there's no money in them from the drug company's point of view. So anything that we can do, and obviously the Gates Foundation, all this is contributing to that because they impact literally hundreds of millions of people. That's something we're interested in. But even in answering the question of what happens when we take a drug, actually, and it may not be immediately apparent, but another big question is, for us, is what's the ancestry and organization of the protein structural universe? And in answering that question, there's actually a relationship to that in the way that we go about answering the question about what happens when we take a drug. It may not be apparent right now, but I'll try and explain to it as we go along. And then part of that also has to do with thinking about, you all see protein structures, you see these beautiful ribbon diagrams, and in many ways I actually call that the curse of the ribbon, because I think one of the issues in doing so is you have a preconceived notion about that protein. Well, let's face it, when a drug is moving towards a protein molecule, there's no way that that drug is going to see it as a ribbon diagram. It's just a conceptualization that's useful to us. But the problem is that we don't actually leave it as such, is we depend on it too much. So it's important to start thinking about how you represent proteins in different ways. So I'm not going to touch on that today, but there's ways of using spherical harmonics and other techniques which actually, I think, take you away from Cartesian space and actually give you a quite different view of proteins, which I think is important. But that's not for today. So our motivation in all this came from when I saw this, which actually, I have to say, blew me away. So this is on the market. These are on market kinase inhibitors, and they've been, Collins and Wokman actually mapped these to the human kinome, of which there are about 500, this is just the phylogenetic representation, there's about 550 kinases in the human kinome. And the size of the spheres here for each of these particular drugs is what they've been shown to binding to and the relative binding affinity. Well, first of all, this notion, therefore, of Ehrlich's magic bullet would seem to go out the window in most cases, perhaps not this particular case, but even with Gleevec and certainly with a natural product like storosporine, it lights up the human kinase tree all over the place. So the idea that you're getting this selectivity is not necessarily the case. And I would argue that it's actually more the norm than it is the exception. And we're going to try and exploit that in what I'm going to present to you today. So I'm not an expert in anything. If anything, I know a little more about structural bioinformatics than I do about these other areas, but each of these other areas is what we're trying to embrace to address this big question. And there's a couple of reviews that have come out this year that if you're interested you could look at. So this is our broad-based approach, and I'm going to describe the algorithmic part, particularly with reference to structural bioinformatics, to sort of get you warmed up. I just want to make a little aside at this point about databases in general. I think the people, even though I'm responsible in part with Helen Berman at Rutgers University in New Jersey with the Protein Data Bank, this is not about us, it's about all those people who work on these projects and biocurators and so forth. In my opinion, these people are the unsung heroes of bioinformatics, and I think we ought to give them a lot more credit than they get. And I think you're using that data every day, and I think you should be grateful for it, and I think you should also make sure you can contribute as much and openly as much data as you can. That aside, I just want to make a point that these things are by far from perfect. And one of the things I've learned about doing research or trying to use my own database to do research is that, effectively it sucks. So, we essentially, there's a research and I guess it's development cycle that goes on, and so we try and do something with a PDB, we can't do it, we improve the PDB, and so the cycle, we do something else, but we then, and the cycle continues. And in this case, the problem I found immediately as I started to look at this is actually finding what drugs are bound to what receptors in the PDB is by no means trivial. And we're actually in the process of addressing that, and that will appear in a release later this year. And just to give you an example, the small molecule drugs and nutraceuticals that are poorly described in the PDB, which is ridiculous. And that's effectively fixed already and be released soon. And then these other stages, of peptides and so forth. And the idea, of course, is when you do all this, everything has to be explicitly and very carefully described. It has to be ontologically represented. And there's a lot of things that go on here, and there's a lot of community involvement and so on. But all of these things are sort of in hand. And I think it'll just make for a richer environment, and it'll interface much better with very important resources to this community like Drug Bank, which is up here in Canada. So that's just a bit of an aside. So what's our approach? What am I going to tell you about? What's the structural bioinformatics that's important here? So what we're doing is we're characterizing, as you'll see, it's essentially reverse engineering the traditional drug discovery process. So what we're doing is we're taking a known protein ligand binding site from a 3D structure or a model, a homology model, or actually a model, and searching for similar sites on a proteome-wide scale, which of course is independent of the global structural similarity or the sequence similarity for that matter. This is actually much different than what happens in a drug company. A drug company will have, a series of assays for looking at this kind of thing. So when they get a lead compound, they'll assay, but the panels that they do are very narrow relative to everything that particular drug could bind to. It's just too expensive to do it. So there's certainly some early stage in the drug discovery process where this kind of thing, if it works, could be useful. And we do this when we find off targets. All right, so what we're doing here is we're finding other sites that have similar binding sites and are likely to bind the drug, which then represent off targets. then we want to actually understand what's the implications of that. And the implications, can be obviously twofold. They can explain side effects of drugs, or on the other hand, it offers repositioning prospects for drugs. And I'm going to give you examples of both of those as I go along. So the applications that we've done lots of things, including some work with Fiona Brinkman here, which I won't talk about today, which actually is just about to be published. I'll talk about repositioning, and I'll look at the protease inhibitor nelfinavir to give you an example of what I mean there. Early detection of side effects is something we're doing with J&J. The drug companies now are clearly in pretty bad shape, and they're actually starting to see the importance of these kinds of techniques, and we're actually working with J&J's two most important small molecule leads. Also late detection, I'll tell you about a case where it was too late, and the effect that $850 million had already been spent over 15 years before the problems with this particular drug, Torcetrapib, was discovered, and what early stage in silico work might have actually done to alleviate that. And then I won't talk about lead optimization, which is another area, but then I'll talk about the notion of drugomes, which really opens up a whole high throughput avenue for actually finding new targets and for finding and repositioning drugs. So those are the applications, but let me just say then, when you actually narrow it down, you actually find them. There are a lot of drug molecules in the PDB, and you'll see exactly how many in a minute. Lipitor is an example. Lipitor just went off patent. It's made over $100 billion in the US alone over its lifetime. But it's sitting there, obviously it's patented, it's in the PDB, and you can go and play around with it. Now, I don't want to imply that there's any kind of real golden solution in here. I'm not going to go into a lot of details of these numbers, but I will just say that so you realize that we're only looking at a small part. The number to really focus on here that's important is the number of structures in it. So here's potentially the number of proteins. In TB, proteome is about 4,000. But we have this many structures, 750 or thereabouts, and from that we can also build a whole series of models. So we only have, still only have 43% coverage in this case, and this is a good case. If you look at malaria, you're down at 25%. If we look at human, we're in the 30 to 35% range. So we're still not getting the kind of coverage we get to get really comprehensive answers, but we're finding some interesting things. The other part of it, of course, is that there is certain bias in all of this, because what exists in the PDB is a misrepresentation in terms of, it's highly representative of certain classes of small single domain globular proteins. So it's underrepresented in obviously what the major class of drug targets like GPCRs. But this is changing. New techniques are emerging in structure determination which are giving us more membrane proteins. So I think I think it's just the classic and beautiful lesson of what we're all doing here is that we're doing much better. Even if we only do the same thing every day, in a week from now you're going to do better because there's more data out there. And if you can integrate those various sources of data, which I'll give you an example of in a bit, you can do some pretty amazing things even if you only have this kind of coverage. So let's look at this, what do I mean by this reverse engineering, and what do we do with it? And a lot of this was developed by Lei Ji in my lab, who's now an associate professor at CUNY in New York, and has just done brilliantly, and he'll just continue to do so. He's just a really outstanding scientist. So what we came up with is, first of all, the idea, and I'm going to give you a little of the bioinformatics of this, is characterizing a ligand binding site in the primary target. All right? That's important because what we're going to do is we're going to search across the proteome for similar binding sites. And we want to know where to look ahead of time. If we have to do an unoptimized search across all possible areas of a protein, across every protein, and say the human proteome, it's not going to work very well. It's just not, it's intractable at this time, at least with the methodologies that we have. So we're actually defining in a very simple way what are the likely areas where the binding sites are, and you'll see how trivial all this is in a minute, and essentially the boundaries of those binding sites. Then what we do is we search, and we use a sequence order independent alignment method for that, and I'll say a bit about that. And then we find things. We find these alternative binding sites, and those alternative binding sites actually have intrinsic ligands as well, typically. And so, of course, the question becomes, well, if we take one of those intrinsic ligands from an off-target, that ligand it should potentially bind to the primary target, just as the ligand of the primary target is binding to the secondary target, in principle. But then what's the statistical significance of that? So the way we've actually measured that initially is by doing some large-scale docking studies and actually permeating and creating standard sets to test with, which was actually statistically very cumbersome and slowed the whole process down. And I'll show you very briefly a statistical approach we're using now, which actually has speeded things up. So let me just say a little about these particular methodologies. So the geometric potential in spirit is sort of like hydrophobicity or electrostatic potential. Is we essentially do a Delaney tessellation, we establish a surface envelope around the protein. And then we effectively look in a very sort of trivial way at what is the environment of each particular surface residue. And in fact, if obviously this is just a cartoon depiction, but if you're actually measuring these distances and the directionality to the envelope, then you obviously get you get these kinds of values are very different than if it's in a flat surface that's exposed to the solvent. And all we're doing here, which is in the spirit of these, is sort of doing this in a local environment, which is what this summation is over a set of neighbors. But it's really just looking at the directionality and distance across the surface of the protein. And it turns out that this is a reasonable discriminator to actually define binding sites. So here's a classic bioinformatics type study where, of course, it's not by any means perfect, and that adds another permutation into the whole system. But it does a reasonable job of discrimination when you have a set of known binding sites and a set of known non-binding sites. You look at the values of the geometric potential. If you do it on a single protein, you have this kind of sliding scale. You can actually pick up binding sites pretty well. So what that's giving us is when we're trying to find a comparative binding site, we're actually homing in on these areas of all these proteins in the proteome to actually look. And it's not foolproof, so we're missing things. There's also a lot of other aspects that have to do with allostery, that have to do with the fact that we might actually be looking at single domains when in fact a drug binding site might be the interface of, say, a heterodimer. All of these kinds of variabilities. But notwithstanding, we still are finding things that are useful. So once we've found the sites that we want to look at, how do we actually compare those binding sites? All right? So here's just an example. With structure A and structure B, and we effectively do an exhaustive maximum weight subgraph comparison. So we're actually comparing all of these particular residues in these two identified binding sites, and the path that we choose is the one of maximum weight. And the weighting is a function of the weight of the nodes. That's where to achieve the optimal path. But what's different in this approach is how we actually get the weighting of those graphs, of those nodes. So you would imagine that if you're doing this kind of comparison, this is sequence order independent. It's not like, yeah.
Could you briefly explain what the nodes represent?
The nodes are C alphas, right? So they're actually, we're taking the properties of the residue at that position. Right? So, and we're comparing those, and clearly if they have similar physiochemical properties, that's one example of why that would weight higher, and that's shown on the next slide, in fact. So you use chemical similarity of the amino acids that you're overlapping in the graph. All right? And then you're using chemical similarity, But I think the thing that distinguishes this approach, and it actually raises, to me at least, a very interesting question, is what we're doing is we're taking two binding sites and we're doing this comparison. But to actually define the best comparison, we're actually using a position-sensitive scoring matrix. So for each of these cases, we have these two structures, but we have a whole series. This is the beauty of it, is that this is where all this data comes in, is we have, for each of those structures, we undoubtedly have a whole series of sequences that can be aligned with those structures. All right? And so we create a position-sensitive scoring matrix. And what we actually observe is within those, we weight the highest where those profiles match the best in those two sites. So it's actually introducing, in some sense, intuitively or qualitatively, you could think about it as introducing a sort of evolutionary constraint on this process. But what's interesting is it a divergent or is it a convergent evolution? Because you can have these binding sites that overlap, but the three-dimensional structures of those proteins are actually very different. And in fact, and certainly their sequences therefore as well. So are these cases where there's been enormous drift and enormous structural change, and so you've got very distant divergent evolution? Or in fact, is it just a matter of convergent, where it's just a convenience factor to have these kinds of residues in the binding site? Obviously, a catalytic triad is the classic example of this, but these are much, much more subtle than that. And then what we've also done is, I won't go into the details of this, but given the time, but we now, in looking at this, we see that if you take a random sample, it follows an extreme, it's exactly the same to what you see in sequence comparison and structure comparison for that matter, a global structure comparison. You can map this to an extreme value distribution and use that as a statistical test. And that's all described in this paper, I won't go into that. So let me just tell you then a little about how we've applied this, because I'm at this point, this was perhaps not what Stephanie and I was wanting to hear, but I'm at the point where the methodology for me is far less important than what you can actually do with it. And so I'm going to sort of focus a bit of the rest or most of the time giving you some examples of what we've tried to do with what amounts to a relatively simple methodology, bioinformatics methodology. So let's look at a couple of examples. So let's look at a repositioning case. Okay? So it's been noted initially in the literature that nelfinavir, which is a protease inhibitor and is used as part of an AIDS cocktail treatment, was noted to have a positive effect, sometimes to the point of complete shrinkage of certain types of solid tumours. So in other words, AIDS patients who are on cocktail treatments who happen to also have these kinds of tumours, it was noted that they often disappeared. And so there was some follow-up on that, and it was noted already in the literature that in fact nelfinavir can inhibit receptor tyrosine kinases, and it was thought that it can reduce AKT activation. So we thought, well, let's use our methodology to try and determine exactly what's going on there. What are the off-targets? And what are the implications of those off-targets? And then there was a clinical trial that was started to actually look at this, well, one could argue more systematically or not, but anyway, there's a clinical trial ongoing associated with this. And so what we're doing here is we're taking the binding site of this nelfinavir bound to this protease inhibitor, and we're doing what I already described to you. We take the structural proteome and we try and map that binding site across the structural proteome. I'll give you some detail on that in a second. But what we're also doing is you've got to realize that these proteins are not static. So the idea of actually just taking the representative in the crystal structure and saying, all right, if it maps to that, if those binding sites are the same, then that's good. If it's not the same, then there's no comparison. You really have to look at an ensemble of representations of that structure, and we use molecular dynamics to that to get the full range of what might be possible. So that, of course, also applies to the ligand itself. And then what we try and do is we construct the network based on those off-targets to see whether we can understand the physiological implications of that off-target binding. So in this case, what we did is we say, okay, we have about 6,000 structures that cover about 30% of the human proteome. Remember the limitations I mentioned earlier. And then we're using the software that we use to do this is all embodied in something we call SMAP. And in fact, you can download it and you can actually use it. It's all open source. And then we have the statistical measure of the comparison of the binding site as embodied in a p-value. So what we found is that just going across this 30% of the human proteome, there were about 126 structures that had significant p-values indicating that they were potential off-targets. When you look at these, you get lots of things you'd expect. You get promiscuous things like P450s, of course, but you also get things like, in this case, the 7 most top ranked were actually other skeletal proteases, which you'd expect. And then, but of the remainder, they were dominated by protein kinases. And in fact, if you increase the stringency of the comparison of the binding site, then 14 out of 18 of the top off-targets are in fact protein kinases. And when you map these to the tree, it's actually kind of interesting because you can actually see almost this in and out motion towards the clades of the phylogeny when you actually increase and decrease the stringency of the comparison. But what's clear here is that there is, what appears to be some significant off-target binding to a number of kinases, particularly tyrosine kinases. Now, all of that's well and good, but what happens when you look at specific cases? Well, epidermal growth factor receptor kinase, what we see is even though, so this is EGFR with an inhibitor bound. This is another structure in the protein data bank, and you can see that this is the inhibitor here, and here's the nelfinavir. Well, at first glance, this is quite different. Well, on the other hand, when you start looking in more detail at the particular interactions, you actually are 74% of the binding site residues are comparable, and you're seeing the similar interactions. You're also seeing things that are extremely important to the inhibition of EGFR. So, for example, this MET793, it forms this hydrogen bond. Without that, you essentially, the inhibition is very minor. So you've got that obviously in the case of this drug, but you're also seeing it with nelfinavir. This is just one example of some sort of detailed analysis of how nelfinavir fits into this particular kinase, and obviously we've looked at others in the same way. But this EGFR is clearly one of the most similar in terms of its binding site. Then, so what you can do is this is just a sort of simplified and broken down version of what you might see in KEGG. And in fact, what we actually see is you can then start feeding this into the network. And so each one of these yellow, with EGFR being the most prominent, are actually identified off targets. And in fact, what you actually see is a slightly different pattern than what was first thought by virtue of this particular off-target binding. I'll explain exactly what that is without getting into the details of the network. But I'll just say here that there was when you start in hindsight going back and looking at some of the literature, there are anecdotal references in the literature that support this kind of activity. So to sort of summarize that, what we have here is the appearance that the nelfinavir, which was this protease inhibitor for AIDS treatment, is actually a broad-spectrum low-affinity kinase inhibitor. And it affects not specifically the AKT pathway, but it's actually upstream of the AKT pathway. And so it actually is a different, pointing to a different place than was originally thought in the literature so far. But obviously these, I should emphasize that all of these experiments, and this is actually some of the nice things that we're doing with Fiona, are our hypotheses without this experimental kind of validation. And in fact, some of this is going on, but that it needs to be supported with more experiment. So let's look at a different case. Let's look at Torcetrapib. I mentioned that before. Torcetrapib is a cholesterol ester transfer protein inhibitor that has the effect, it's cholesterol control. It was going to be Pfizer's follow-on to Lipitor when Lipitor went off the market, went off patent, sorry. And in fact, you know, what happened is that the CEO of Pfizer got up and said, you know, this is coming, it's in stage 3, everything's looking good. Their shares started to go up like that. What actually happened is that literally a week or two later they had to withdraw it from stage 3 clinical trials because there was a number of deaths that were attributed to hypertension. So, you know, this is something that had taken 15 years to get to this point, and it had $850 million spent on it. So, you know, the idea, of course, is to fail early and fail cheaply. That's not what happened here, but that is ideally what you would like to do. So we decided in hindsight to take a look at this, and we sort of done it in a different way. I'm not going to go into the details. We found about 12 off-targets in the same way as I showed you with the Nalfinavir study. These are very different off-targets. What's interesting in that case, that there's a number of nuclear receptors as off-targets. But what's interesting is that there are follow-on drugs to Torcetrapib, and they have different binding profiles, and those different binding profiles actually map to different physiological outcomes. So that's, you know, an interesting anecdotal indication that these would appear potentially to be true off-target binding sites. But what we've done in this case is, what I wanted to tell you here is a sort of different study that was done by Roger Chang. It's sort of coming in the other direction. And it's using, and Roger's a joint graduate student with Bourne and Paulson and myself. And what we used there are some of Paulson's metabolic models. And what we tried to do is essentially you take the metabolic network reactions and you represent that as a matrix representation. And then you assign, or you try and assign relative, you assume a steady state, and the idea is to assign fluxes to that, and you actually have a flux space. But when you actually you exert a drug into the system, then you actually perturb that drug space, that flux space. And what we try and do is to interpret that, and we've done that by using because this was a hypertension study, it turns out they have a renal model, they have a human renal model, which is actually very good for the point of view of measuring hypertension, because a lot of the activity, a lot of the features are embodied in the model. And it's, again, by no means complete. But there's a whole series of pathways, reactions in 7 cellular compartments. And this sort of, the idea here is to bring a whole series of data together. So you actually have this preliminary model, you have these constraints that you put on the system, you then bring in additional data, objective functions from the literature. In this case, there's also some gene expression data, and you look at that in the context of the model. And what actually comes out of that is some causal drug off-targets that are established through the model. And in doing that, then you look at those compared to the off-targets we get by the methodology that I described to you, the structural bioinformatics methodology, and you see what kind of overlap. This is incredibly preliminary, but I think it points to what might, as these models get better, then I think the idea of determining causal drug off targets through these kinds of models has some promise. And when you bring these methodologies together, it's sort of almost like a consensus-based approach to actually understanding what the off targets are and what the implications might be. So that was just another study. And I'll just give you one example of a sort of high throughput approach to this that was done by Sarah Kinnings, and I'll just use that to sort of finish off with. But the idea of doing this in a very high throughput way, with the idea, this is just back to the structural bioinformatics part of it, with the idea of identifying a whole series of new potential targets within a given proteome, and the idea of identifying particular drugs, particularly, of course, it would be off-patent drugs would be ideal, but drugs that are already out there that you could reposition to treat these kinds of conditions. Obviously, there's a huge advantage in repositioning from the point of view it's already been through the FDA, and well, at least in the US process, and it really can be short-circuited. Other trials have to be done, but it can reduce the time to market from 15 years to like 2 years. So just looking at tuberculosis as an example, what we did here is we computed the TB drugome to actually get at these additional possible targets and drugs. Now, this is clearly an important problem because there are multiple drug-resistant and extreme drug-resistant strains of tuberculosis, and in fact, the number of those strains is actually increasing. So that the treatments are becoming ever more problematic, and there's a huge number of deaths associated with it. So to compute the drugome, what we do is we determine this proteome, and I'll say a little about that, and then we determine all the known drug binding sites. And I think this begins to speak to the promiscuity of drugs, all right, and how we need to try and embrace that and take it into account. So we take all known binding sites from the PDB of drugs, and we see which of those sites actually exist in the TB drugome, and we call that the TB drugome, and then we try and use that information. So it's a sort of large-scale study. So if you want to determine the TB structural proteome, it turns out if you go to the PDB, there are only 284 structures. But with modeling, you can actually get that number up to about 1,400 and something, which is about 43% of the 4,000 that exist in the whole proteome. So again, not great coverage, but not bad. Then you take the drug binding sites, and so we took just FDA-approved drugs in this case. And already this speaks to the promiscuity of those drugs. So there's 268 drugs which are bound to 931 binding sites within the PDB. We're not talking about things that are highly homologous. We're talking about things from very distinct gene families. All right? So for example, and this is no secret, but something like methyltrexate is a very promiscuous drug. That's actually bound to 37 different distinct targets in the PDB. And then, there are others. Obviously, there's a lot that are only bound to one, but there's certainly significant promiscuity here. When you actually compute this, what you get is you get a whole set of binding sites in blue, and then you get a whole set of approved drugs in red. So clearly, where you have hubs around the red, you've got a particular drug that's binding to a whole series of receptors. This has potential value because if you're actually hitting a whole bunch of targets at once, one has to balance this in the context of what happens in the human drugome. But in principle, if you're hitting a whole set of targets at once, the chance of getting a mutation and an adaptation and a new strain is less likely. You're just knocking it out. So these obviously have some potential value, and you can actually look at it in the other direction as well. So just to give you an example of that, so the similarity between the drug binding sites and TB proteins, so there were 61 drugs out of the 268 that had binding sites that mapped to TB TB proteins. And 41 of these drugs actually bind to more than one TB protein. So here's just examples of what that looked like. And you can see that in one case, it's binding to actually 14 different TB receptors, TB proteins. And when you look at what these these drugs are binding to. They're binding to a whole set of some identified targets. INHA, which I'll say something about in a second, is one of the most prominent targets in TB. But there's a lot of targets here that have never previously been identified as possible targets. And these are obviously drugs on the market. Voxithyroxine is used to treat goiter and chronic lymphocytic thyroidosis. So, there's a lot of indications which are quite different. So, there's a huge wealth of information here, and the question is, how useful is it? And it's a matter of choosing cases. So if I give you an example of cases that we've chosen, and just to give you to sort of put the value of this into some perspective. So entacapone and tolcapone are two drugs that are used in the treatment of Parkinson's disease. They're actually COMT inhibitors. And so they showed up pretty well in this, and so we looked at that. We looked at it in particular because they bind to INHA. INHA is a particularly well-established target in tuberculosis because it's actually involved in the fatty acid elongation cycle, and there is no representation in the human that comes close to INHA. So it's actually been a pretty active target over the years. And so these two drugs, we do all the docking and all the other stuff to it, and they look like pretty good cases. Then we did some in vivo support, and that too was positive. Assays of direct binding, and so all of this looked pretty positive. So we handed this over to the TB Alliance, which is a non-profit organisation trying to deal with TB. The problem in this case is in their current form, so there's good news and bad news. The bad news is that the minimal inhibitory concentration, the MIC, is 260 micromolar, which is actually way too high. The toxicity is going to be problematic. They also have other INHA inhibitors in the pipeline that they're working with more actively in terms of medicinal chemistry. The interesting thing about encapsulone and tolcapone in this regard is in their current form they may be too toxic. But what they do is they have chemistry that's unlike any other current inhibitors against INHA. So it might be that they could actually be good starting points for future study. So these are just examples I've given you of trying to use fairly simple bioinformatics techniques, in this case actually identifying where binding sites might be on proteins and then comparing those binding sites and applying that to actually look at the notion of promiscuity, which is now a popular term of polypharmacology, this idea that either individual drugs or multiple drugs acting on multiple targets is a way to extend how we approach drug discovery. And then what we're just beginning to do is we've done the network part in the sense of static networks where we look at the implications of these off-targets by mapping them, say, to KEGG pathways and looking at what the implications are there. And then I think the future, which is still a little ways away, but the idea of systems pharmacology, we're actually doing this in a sort of dynamic way where we're building these networks and looking at the causal targets from a different perspective. So I think what we're going to see, and given the current state of the drug industry, I think there's a certain interest in embracing these new approaches to drug discovery. And clearly computation is going to play an important part. So I think I mentioned everyone as I went along. I didn't mention Jan, but we didn't discuss his work. So I'll stop there. Thanks very much.
When you calculate the p-values for your binding site, is this based on the potentially biased set of known protein binding sites?
Yeah, there's actually a database of known non-binders. It's called DUDS.
Okay.
So yeah, we use.
Negative set.
Yeah, That's an established negative set, so we use that plus our positive set.
And so for the drug repositioning, so in the first part, you talked about find a similar binding site for drugs. So it's conceptually, I think it makes sense. But if you think about the drug protein docking now, it's very fast. You can dock within like 10 seconds. So you actually can dock the drug to all the protein you have. So what's the advantage you just dock to your similar protein?
Well, I think if you take all the binding sites, I mean, you want to do it accurately, and you want to do, you don't want to just do the one instance of that drug. You want to do, and this is what we do when we use this methodology essentially to pre-select, to then do what you described, because what you described, docking is fast, but the faster you do it, the less accurate it is. Plus, you're talking about one member of what really ought to be an ensemble of docking. So, in other words, where you take a simulation, you might do 300 or 400 different dockings to that particular protein in slightly different conformations of members of that ensemble. And then you can use autodoc Vena, and you could probably, on a large cluster, you could even do that. But the idea here is to home in on things that are actually more meaningful to start with. But once we actually so we use this essentially as a pre-filtering to then do, for the cases that look the most promising, to do a more extensive docking in the way that you're describing, but not across the whole proteome.
Yeah, because I'm thinking no matter how good your how good your program can really find similar pockets, you could miss some structures, right? It's actually a similar pocket, but miss them. So if you dock everyone,
but you can't do random docking on all potential areas of a protein that might actually dock that ligand. You have to have some form of filtering. I don't believe that problem's anywhere near tractable at this point, not on a proteome-wide scale.
I guess it's a general, maybe naive question about the binding. Is it always the case that if you have a ligand binding to a protein that it's antagonistic to the protein's function, or is there many incidences of binding that are simply incidental and inconsequential, in which case it really wouldn't matter if the drug bound to those members of the proteome? Maybe related to that, what's to prevent someone from just doing a high-throughput experimental assay on the FDA-approved drugs with TB and just trying every one of them out and seeing which ones actually were deleterious rather than inconsequential?
So, for your second question, I'm not an experimentalist, but that's not something that drug companies are doing. In fact, drug companies aren't necessarily even interested in looking at any of this TB stuff. I mean, we've approached Novartis and people. There's no money in it, right? And they're not doing this kind. I mean, I can't. I guess we're on tape, but drug companies strike me as like super tankers. That getting them to stop or turn is very difficult. That the processes that they use, that they've used for a long time, they're very vested in those, and by virtue of the fact that things aren't doing well, and they've laid off lots of people and they've reorganized around diseases and things like that, I think we're starting to see change. But overall, this is not the kind of thing that they've embraced, which seems to me, which is what I said right at the beginning. It all seems a bit crazy to me.
And the question about incidental binding versus...
Yeah, I mean, I think there's undoubtedly, lots of that, and it's, how do you distinguish? I think if there's significant binding, it's, I mean, just anecdotally by virtue of experimental support that we've got. I mean, I'm only outlining, we've had, I don't know, 20 papers of different case studies of this, say of which, I don't know, 10 have been experimentally validated. So I think by taking the most, the cases that show the greatest similarity in the binding sites, and then when we do the docking scores are problematic. In fact, we've developed, I didn't talk about this at all, but we've developed some machine learning approaches to actually try and improve docking scores. But if you're getting a reasonably significant docking, then I think, you know, it would appear that these are reasonably significant binding event or interaction events versus, you know, I mean, there's clearly an enormous amount of transitivity in the whole system, not just in terms of ligands interacting with proteins, but with proteins interacting with proteins. So, you know, I mean, we're just working with what we have. But I mean, it's a valid question. Okay.
You mentioned levothioxine as a very ubiquitous binder. I think it was 14 potential binding sites. So, but it's also, as far as I know, naturally occurring. I mean, something that the healthy body would make anyway, right? So, is it then necessarily a problem that it is an ubiquitous binder, or is it just the problem that if you take it as a drug, that it's in the wrong place when it's making these interactions? Is that the main concern there?
Yeah, I think partly, but you've actually triggered something else in my mind about the notion of effectively not necessarily natural drugs that the body produces, but natural products themselves, right? That the whole drug industry went, you know, into this notion of using natural products or trying to design based on natural products, and that there is no, you know, that just did not work out. As far as I'm aware, there's no major drug company right now that's actually looking directly at natural products as drug molecules. The idea is to design things that take elements of natural products. Is the direction they seem to be going into.
And the definition of natural product is it's something the body is making itself, right?
Well, it's something that the body is quite comfortable with by virtue of some coevolution or.
Because I think, I mean, as far as I know, if this is the case for levothyroxine, it's something that the healthy body would make anyway. So if then the interactions are concerned, it would be because it's the right thing but in the wrong place in the body.
Yeah, I would assume so. I haven't really looked into that case in particular, but you actually raise a good point.
And then I saw, and this was one of your outliers at I think 37 or something. This is a really common drug in chemotherapy, like mesothelioma.
Yeah, leukemia.
So if you look at, say, the top list of what it's interacting with, did this raise any red flags? Or because I know it's not just used for treating, you know, cancer, it's used for treating arthritis and whatnot. And this is not what's in the small print, you know, if you look into the package.
Right.
So did you see any interesting things when you?
No, I mean we haven't really looked into a lot of this massive data, unfortunately. And you know, I think there's a lesson here for the group, and I should take it myself, that you know, just throwing a large amount of data out there doesn't really get, you know, you're not going to get the attention of people. You know, so what we've tried to focus on more and so these drug OMs, I think, are just the beginning of actually beginning. We need to focus these cases ourselves. We haven't done that yet, because, you know, just having a massive drug OM. I mean, there are some interesting bioinformatics aspects to this. Is, you know, what is the characteristics of these drug OM networks? Are they scale-free? You know, these kinds of sorts of questions, which we've sort of been looking at a little, but not the actual implications of each individual, you know, hub or within those networks, which is, you know, something that really needs to be done. Because you're raising very valid questions that we haven't really looked at. I guess it's beer time.